Okay, welcome everyone. Thanks for joining us today to talk about FAIR data principles and open science. I'll start this event by acknowledging that I'm joining you from the land of the Gadigal people of the Eora Nation. I also acknowledge the traditional custodians of the various lands on which everyone attending virtually today live, meet and work, as well as any Aboriginal and Torres Strait Islander people in attendance today. I pay my respect to elders past, present and emerging and celebrate the diversity of Aboriginal peoples and their ongoing cultures and connections to the lands and waters. My name's Juliet. I'm the Atlas of Living Australia, or the ALA's Training and Outreach Coordinator, and I'll be moderating the session today and facilitating a Q&A we've set aside time for at the end. So if you do have any questions while our lovely speakers are talking, please pop them in the Q&A box at the bottom and we'll get through as many of them as we can. This is the second of 3 events for National Science Week that we're running at the Atlas of Living Australia. Tomorrow we'll be running our last event, which is about ensuring citizen scientists provide the best quality metadata they can to support their observations. We're joined by 3 panellists from the ALA today, each of which will be touching on a different element of the topic. I'll introduce each of you, then hand over and we can dive straight in. So Dax is here. He is a data analyst at the Atlas and the science lead in the science and decision support team at the ALA, where he tries to make it easier for people to use ALA data for robust and transparent science. Dax is the co-maintainer of the Galar R package, and he also has a PhD in evolutionary biology and social psychology from UNSW in Sydney. Ely Wallis. Ely leads ALA's engagement team. She also works closely with the museums and herbarium collections community. Ely is the current chair of Biodiversity Information Standards, which is an international organisation that develops standards for sharing biodiversity data, which is work that underpins sites like the ALA. Prior to joining us in 2018, Ely worked for over 20 years in Museums Victoria in Melbourne, where she spent time in collections management, got the Biodiversity Heritage Library project up and running in Australia, and developed collections, online websites, apps, and in-gallery multimedia experiences. And we also have Peggy Newman here, who is ALA's data manager. She has been with the Atlas for 9 years and started as a developer on the ZoaTrack platform for animal tracking data, which perhaps some people here are familiar with. Her long experience with data warehouse management led her to this position. And the data team here work with our data providers all over Australia to harvest and ingest data into the ALA's data pipelines. They also work on building the ALA's taxonomic backbone, maintaining conservation lists and the sensitive data service. So yeah, to start off with, I'll hand over to you, Peggy, if that's okay.
Great, thanks, Juliet.
No worries.
Will just have a go at sharing and we'll see what it looks like. How are we going? Can you see my screen? Great,
yeah,
okay. And I'd just quickly like to do an acknowledgement of country for Naarm in Melbourne, where I work and live. I'd like to acknowledge traditional owners, the Wurundjeri people of the unceded Kulin Nation, and pay my respects to elders past and present, and extend that respect to all Aboriginal and Torres Strait people today. So I'll be talking about the FAIR principles and how they're implemented in ALA infrastructure. So in a fairly practical way. The Atlas of Living Australia, as I hope much of our audience will know, is Australia's national biodiversity infrastructure. Biodiversity data infrastructure. So we speak in occurrence records, so a species in a time, in a place. We've got lots of records, we've got lots of data partnerships, registered users. Our infrastructure is open source software, and it's used by 27 countries around the world. We've got a lot of international relationships. The main one we'll be talking about today is that with GBIF, with the Australian node of GBIF, which is the Global Biodiversity Information Facility. So we're talking about FAIR today, and I guess I wanted to mention that we're not just doing It's a key principle underpinning the NCRIS funding that forms a large proportion of our funding. So NCRIS is the National Collaborative Research Infrastructure Strategy, which states that data generated, created, captured and stored by NCRIS projects will be made available to the wider research community based on FAIR principles. So it's a really important part of our funding and therefore really an important part of our work culture. So what is FAIR? FAIR is an acronym, obviously, findable, accessible, interoperable and reusable, with this short and succinct bunch of actionable guidelines for how you can make your systems and data fair. Not so short and succinct that we will run through all those today, though, so I've prepared a summarised version. More simply, we can talk about findability in terms of data having rich metadata and unique identifiers. Accessibility can be described as the data being easily downloaded by using standard protocols. Interoperability is where metadata is accessible in standardized language, and reusability is where data is well described and provides clear usage and licensing. So I'm going to broadly cover these topics one by one, citing some examples in our infrastructure as we go. So starting with, just a moment, and I can get my slides moving properly, facet searching. So starting with findability, one of the key ways that people navigate the ALA is through what we call facets, and we bring data into the ALA and we standardise it into Darwin Core and into fields and vocabularies that we index, and that allows searching quickly via facets and APIs. So in this screenshot here on the screen, you can see we've got our main interface for navigating occurrence records, and you can look at those records in tabular form, or you can look at images, or here we've got a map interface. But on that left-hand side, under narrow your results, are our facet groupings. And we mostly explore occurrence records by taxonomy and by location, but there are other dataset type facet groupings there as well. So those are the main ways that we search the ALA. Taxonomy is the main one, and I wanted to give this special mention because what we do is we go to really quite some effort to work to create a taxonomic names index. By combining a whole heap of authoritative sources in Australia and their classifications. So what that ultimately means is each occurrence record that comes into the atlas may only have a limited amount of taxonomic information provided with it, and we will augment each record with everything that we know about that taxon. So here you can see in this screenshot, we've got the original versus process table. You can see that a data provider has only given us the scientific name for a koala, but we have augmented the record with everything that we know, including the identifiers from the authoritative sources, in this case, the Australian Faunal Directory. This helps us do a lot of taxonomy processing and searching and indexing in the ALA, and it also helps us with doing other things like flagging threatened species and sensitive species. Similarly, with spatial data will come in coordinates through the data provider, and we will do a whole heap of spatial parsing on that record and then check whether we can map it or not, whether we think there are any errors in it, and we will sample that set of coordinates against around about 120 spatial layers. So we have polygonal layers and environmental layers such that each occurrence record can then be classified by one of these spatial covariates. That helps with searching as well. Also under findability, we talk about identifiers, and we've got a few global identifiers that we use and talk about in the ALA. We mint our own identifiers against each occurrence, which it provides consistency in our own system, and also provides a globally unique identifier for each occurrence record. Museums and collections maintain institution codes and collection codes and catalogue numbers for each of their preserved specimen, which we document in our metadata registry. On an international level, GBIF maintains internationally similar indexes. GR-Cycol and the Index Herbariorum are registries for international collections and institutions. Also, GBIF mints DOIs on interpreted datasets as another globally unique identifier, and it manages the linkages between identifiers. And I'll talk more about those DOIs on datasets later on. Moving on to accessibility. Accessibility is about being able to access data and download data. We have our user interface that allows searching and exploring data via species and location again. So we have a species search when you go to the ALA and the main page. We have a search box that searches our names index. Species pages to explore species information, and we've got a lot of different mapping tools, including Explore Your Area and our spatial portal. But the ALA is what we call a service-oriented architecture using open source technology. So what that means is that we have all of our front-end applications, but our back-end is those front-end applications are serviced by an API level, an API layer. So that serves up all of the data. Now that API layer is actually free to use to our users as well. And you can see here, you can access services that will just serve data for occurrences, species, metadata, spatial services, etc. So for those of you who aren't familiar with APIs, it just means that somebody can get all the data without having to go through the web page. Dax will talk later about our R and Python Galar packages and how they interface with the API, so they make it a bit easier to query the API using those packages. Authentication and authorization are an important part of the FAIR principles because you can't just willy-nilly release data, especially if there are some sensitivities around it. We have a sensitive data service in the ALA that obfuscates coordinates for sensitive species as directed to us by state jurisdictions. We manage the access to that data, and we also have a recent project called the RASD framework, the Restricted Access Species Data Framework, which provides a legal framework and workflow for granting access to sensitive data. Moving on to interoperability, I, as a data person, see two types of interoperability, or talk in terms of them. Syntactic interoperability, first off, is where we use data standards and communication protocols to move data between organisations. So for us, for our metadata registry, we use the EML protocol, or ecological modelling language. That's for describing data set level metadata, I guess. In terms of data, we use Darwin Core. So Darwin Core is a standard, or a bunch of standards, primarily based on taxa and their occurrence in nature as documented by observations, specimens, samples, and related information. So Darwin Core tells us what terms we can use in our files and in our data. The format that the ALA speaks is a Darwin Core archive. So, and that's put really simply text files, self-describing text files wrapped up in a zip file. And that's the primary format that the ALA and GBIF uses. We both input and output those formats, that format. Then we can talk about semantic interoperability, and that's less the type of vehicle that you use to move the data, but making meaning of the data so that it makes sense. Vocabularies are important for semantic interoperability, but they're also important for discoverability because they help us organise the data into those facets I was talking about earlier. So Darwin Core, some might call a vocabulary where it's a list of terms governed by TADWIG, so it helps us understand what those terms mean. And if the ALA gets a term in our data core basis of record, we understand what that's intended to mean. TADWIG is the Taxonomic Databases Working Group who maintain the Darwin Core Standard, and we're having our conference in Hobart later this year. Between TADWIG and GBIF, there are some controlled vocabularies for particular terms, as a few examples I've listed there: basis of record and establishment means, the current status, etc. There's also vocabularies being prepared for systematic survey methodologies in what we call a Humboldt extension in the TADWIG world. But vocabularies are becoming more and more important in our data landscape. Moving on to reusability now. I think Ellie might talk a lot more about Creative Commons licenses a little bit later on, but in the Atlas, well over 99% of our data sets are, occurrence records are available via some form of Creative Commons license. So I've taken a screenshot here of the breakdown of figures. We've got a lot of records available with a completely open license, and there are all sorts of different permutations of license there. So just something I'd like to point out internally for us is that the systems are set up such that you can have licenses against an entire data set at the occurrence level or even at the image level. A really important thing about reusability, I think, is that the data provider is able to see how their data is being used. So the ALA provides some logging or some usage tracking for data providers to be able to go and see how their data is being used. So this is an example for Frog ID from the Australian Museum. We can go to their page in our metadata registry and see that just this month, for example, Frog ID records have been downloaded in over 1,000 downloads. And when people do a download in the ALA, we ask them to give a reason why they've downloaded. So we can see here that Frog ID is being used extensively for scientific research, for example. Another way that this kind of usage tracking is played out in ALA and GBIF is that comes back around to what I was saying earlier about GBIF. When a data set goes to GBIF from the ALA, a DOI is minted on that data set. So Frog ID, some time ago, was loaded in the last couple of months. It went to GBIF. GBIF mints a DOI on that data set, but it also tracks citations on that data set. So both the ALA and GBIF track mint DOIs separately on downloads. Each and every download that comes out of our systems is DOI'd, and then GBIF maintains a citation tracking service which will track GBIF downloads, and it will actually do a matchup and allocate the citations back to the dataset download. So that results in a bit of a count, some analysis, and you can go to GBIF and download a report and see how all of those, how that data was used and the publication details. So that's it for me. I think I can answer questions that anybody has. I think that was fairly quick and covered a lot of things. But thanks very much, Juliet. I'll hand back to you.
Cool. Thanks, Peggy. That was really interesting. I think we'll do all the questions at the end, just so we can give everyone time to digest all of this information. So I'll hand over, I think, to Ellie, who's next. And just check that you can get your slides up and everything, Ellie. Perfect. No worries.
And unmuting myself might help as well. Okay, hello everybody out there. We can't see any of you, but it's great to have you join us today. My name's Ely Wallis and I work as the Engagement Team Lead and Collections Community Engagement Manager here at the Atlas of Living Australia. I'm talking to you today from Wurundjeri Woiwurrung country of the Kulin Nation, and I pay my respects to elders past and present and to any Indigenous people attending the meeting with us today. As you heard from Peggy, the ALA has a bit over 125 million occurrence records supplied by over 850 data providers. The cumulative number of records in the ALA has grown steadily over time as more data providers have come in and new records have been added by existing providers. But the spread of the records isn't even across a few different dimensions, and one of them is the types of animals and plants that have records. As you can see from the size of the dots on this chart, birds easily make up the biggest number of records within the ALA, followed not too far behind by vascular plants, and other groups have a smaller number of records, even though numerically out in the wild they might have more species. The data are also not spread evenly in terms of geography or data source. So on the left, the map of Australia shows the unevenness in geographic spread. So the darker the orange colour, the more records are held. But this is not all that surprising. The population in Australia is much more dense around the coasts. So that's also where the collections have been made. The centre of Australia is a sparsely populated desert with few roads and difficult terrain. So it's not all that surprising that fewer collections and observations are made there. So the spread is also not even when you look at data source. Now on the right, this is a very simplified graph which shows the date of collection or observation against the number of records. Only two data sources are shown. The dotted line at the bottom is for museum and herbarium specimens data, and the line going sharply upwards is for human observation data. So the human observation data that we get into the Atlas is from big citizen science projects like iNaturalist. And also, if you remember that birds are the most recorded types of species within the Atlas, there's really big bird observation and monitoring programs that we receive data from, like eBird and BirdLife Australia. So the easy takeaway from this graph is that the number of human observations has increased dramatically since the early 2000s. And this coincides with the rise of citizen science and has only continued to increase with the popular use of smartphones that allow good photos and accurate geolocality, date and time information to be recorded. The collections records obviously plateau off, and it's easy to take a bit of a lazy view of the plot and wonder why the collections are not keeping up. Well, of course, if we were collecting things and putting them in museums and herbaria at the same rate that the observations are being made, then there wouldn't be much left to observe. And of course, there are hard limits within museums and herbaria on storage capacity and on the capacity for the collection staff to deal with all the material received. What I haven't shown here are two curves that might eclipse both of those curves that are on the graph at the moment. And that's for machine observations and for eDNA, two new types of data that we're now really being challenged with within the Atlas and within GBIF. So that gives us a 10-second snapshot of the types of data that come into the ALA. But if I want to use that data, are there biases in there as well? I'm going to take you back to a slide that Peggy's already shown when she was talking about reusable as one of the FAIR principles. As she explained, the table shows the number of occurrence records that hold a Creative Commons license. And I thought it might be worth digging into Creative Commons a little bit more. So let's start with what is Creative Commons? Creative Commons is a not-for-profit international organisation that provides a legal framework for sharing creative works. It's underpinned by copyright law, and that's really important. So Creative Commons only exists because we have copyright law. But Creative Commons provides more than a simple in-copyright mark, where the in-copyright mark doesn't really tell a prospective user anything about what they might be able to do with that creative work. If the creative work is a photograph, a piece of media, something written. Simply saying in copyright also doesn't provide the copyright holder with any way of letting people know what the copyright owner is prepared to allow with the works that they've created. There are 6 different licenses with varying degrees of openness, and I'm mostly going to be talking today about CC BY, the attribution license. That's the least restrictive license within Creative Commons, and it only seeks that the creator of the work be credited for their work. You can see in the photo of the barnacles that the attribution can be very simple, and it can be nothing more than the person's name and the license type. So, as Peggy already pointed out, many records in the ALA are licensed with a CC BY license, and we'll return to that a little bit later in the talk. But there's another license type that's also worth mentioning in the list, the CC BY non-commercial license and the other variants that use a non-commercial license within it. There have been many debates about what is commercial reuse versus non-commercial reuse. Because it's still a grey area, some sites actually won't accept images or creative works at all that carry the non-commercial clause, including Wikimedia Commons. So if you ever want your creative work to appear in Wikipedia in any form, then don't license it with any of the last 3 licenses. They won't accept it. In fact, don't use a non-derivatives license either, because that won't be accepted either. Now I'll move on to a couple of additional sort of non-licenses before returning to CC BY. The public domain mark is used for works that are no longer covered under copyright law because they were produced a very long time ago. The date at which a work enters the public domain varies around the world depending on different jurisdictional copyright law. But a publication like this, which was made in 1802, is very clearly in the public domain. And this work that I've put up here has been, I've taken it from the Biodiversity Heritage Library. You aren't required to provide an attribution for works in the public domain, but it's still good practice to do so. The Biodiversity Heritage Library project is also working hard to add DOIs to historic literature so that the historic literature is more easily citable in modern publications. But that's the topic of a whole other series of talks. And if you haven't heard about the Biodiversity Heritage Library before, I'd really encourage you to go looking through it. There's 61 million pages of digitised literature, and it may well help your research in ways that you can't even imagine, as well as having beautiful illustrations to look at along the way. The other sort of non-license to mention is CC0. There are about 42 million records in the ALA that are licensed as CC0. This license, or kind of non-license, is used widely to declare that the content is in the public domain and there's not even a requirement for attribution. And that's great because it makes it super easy to reuse those records in further scientific research. eBird, as one of the biggest data providers to the ALA, have adopted CC0 for data that they're sharing to the ALA. And so you can see from the map showing the eBird data points that gives users vast amounts of data with excellent geographic spread already available for reuse with no need for attribution. Another example of an open science site that has adopted CC0 is Figshare. Figshare is a data repository that allows researchers to publish their research data sets in a citable and shareable manner. When a researcher downloads data from the ALA and conducts their research and then publishes a paper, best practice is to upload that data set into a site like Figshare, as it makes the research more reproducible. And reproducibility is what Dax will be talking about in depth next. So let's go back to the breakdown of licenses used on data in the ALA. The very top one is CC0 with a bit over 42 million records. The second most common type of license is CC BY, the license that asks only for attribution. There have been a number of updates to that CC BY license over the years, which is why you get variants coming in, which is shown with those light blue arrows. CC BY put onto the data is often thought to be as providing the least restriction on reuse. And it is certainly the least restrictive of the 6 official Creative Commons licenses. But as we've seen already, using CC0 on individual data records is actually better for reuse. Imagine trying to produce a scientific paper where you had to credit every single data point. People generally just don't do it, and the default becomes to attribute the data providers as a whole, which sites like ALA and GBIF make easier to do by creating the DOI when you're downloading records. And the DOI on download is something that Peggy mentioned in her talk. So when you cite that DOI, you've also cited the contributors. So that's not to say that there isn't a place for CC BY licenses. CC BY licenses are very commonly used on documents, and you'll regularly find the CC BY mark on government reports and that sort of thing. An example is the ALA's strategic plan. It's publicly available on the ALA website and can be reused by anyone without needing to seek any explicit permission from the ALA. All that's asked for is attribution. That's the BY bit of CC BY. And in the case of the strategic plan, ALA tries to make it easier for the user by providing some guidance with the citing this publication text. But you didn't come here to hear about a strategic plan. It's really the data that we're interested in and how the licenses that apply to that data help or hinder the reuse and open science. So records from Museums Victoria give an example of what's really considered to be best practice and how they really understand the uses of the licenses. So this is a species occurrence record, the nuts and bolts of the ALA. And on this occurrence record, we can see that on the right-hand side in the blue sort of rectangle is the license for the data. And the data, so the data, the text record of the data is licensed as CC0. On the left-hand side, or you can see there's a picture of the little critter, and the critter's image is licensed differently, and it holds a CC BY license. So that's helpful because it means a researcher who wants to use the data as part of an analysis or as part of a data set that they download for modelling or something, they're free to do that. But the image, which is much more likely to be used individually and was created less ambiguously as part of a creative process, so it's very clear there was a creative process involved in taking the photo, is asked to be attributed with a BY license. Consistency across sites is really important when it comes to this sort of thing. So this is now Museums Victoria's Collections Online site, and you can see that the data and the image are licensed in the same way as they are on ALA. So you really want to make sure that you're consistent across sites. So the message is, make your data as open as possible, and CC0 is an excellent choice. For images and other media, the least restrictive license is CC BY. And if anyone is worried that putting your images up with a CC BY license will mean that others will take the images, put them on tea towels and sell them down at the market, well, yes, that is legally allowed. But don't forget you can sell them too. So if it's a genuine concern that other people are going to sell your images, then make sure you've got your value-added merchandising on offer and you can enjoy the best of both worlds. Looking for the last time at the spread of licenses in the ALA, I've now highlighted those records that would be considered to have restrictive licenses, and they're highlighted with the orange arrows. And particularly that's the non-commercial use licenses. What constitutes non-commercial use, as I said earlier, has been the subject of many debates, and some sites like Wikipedia simply won't accept non-commercial licensed content for reuse. With reusing data points from the ALA, we can imagine a scenario where what defines non-commercial or commercial reuse is complicated. So we have industry users of the ALA, such as environmental impact assessment companies, and those industry users are profit-making companies. They take and are allowed to take freely available data on the ALA and use that data as one of their inputs into the research that forms the basis of an environmental impact assessment and the recommendations about development approvals. But if we took the line that restrictively licensed data should not be available to these users because they're working for a profit-making company, then those users would not have such a good data set for their downstream research. And deprived of that bigger data set, which might have really important data in it, the companies may end up drawing different conclusions about what the data is telling them, leading to different recommendations about the impacts of development on the environment. So I'd ask the providers putting non-commercial licenses on their data, why don't you just put an open license on it? Let anyone use it, because the downstream benefits may not be obvious, and what constitutes non-commercial is also not always obvious. In the last couple of minutes, I'm just going to quickly touch on a couple of additional issues around data licensing as it relates to FAIR principles. Sometimes when it comes to seeking permission to use data, images, or information, copyright isn't actually the problem. Instead, what we need to look at is our social license to reuse the information or the data. The benefit to community, ethical behaviour and seeking permission are what's needed. And these ideas are encapsulated by the CARE principles, a different set of principles but related to FAIR. The CARE principles were developed by the Global Indigenous Data Alliance and speak to issues around data governance and Indigenous data sovereignty. They were drafted in 2018 by an international group who met together in Botswana. An example of where we might need to apply the CARE principles in relation to biodiversity data is where we've got Indigenous knowledge, such as language words or descriptions of how a particular species was used, if it had a medicinal use or if it was a food source, actually written on herbarium sheets. And there's a very active debate and attempts to try to work out what's best to do with that data. If you take a photo of that herbarium sheet and put it up online, then that data is there for all to see. But that might not be acceptable to some communities, and trying to work out what's the best thing, whether we should find the community and ask them first, or whether we should put the data up so the community knows that it's there, is still something that the herbarium community are working through. So I'd encourage you to find out a bit about the CARE principles, as they're very relevant to new field collecting, to co-designing research on country with Indigenous communities, and changing our perspectives to encompass Indigenous worldviews. So finally, I'll leave you with a few thoughts about how open science is an international agenda. Open data is not just about biodiversity. Open data and open science is being discussed in many forums. It's not, and there's a few areas of open science and open data that I haven't even touched on today. For example, open access publishing and public engagement with science. But open science benefits us all. So when you're thinking about providing access to the data, think about just how open you can make that data. Does a restrictive right license really make sense? And if it doesn't, then be bold and go open. And make sure that you can consider the CARE and FAIR principles while you do it. Thank you very much.
Cool. Thank you, Ellie. I think we're going with Dax next.
Yeah, hold on. Great, thanks, Juliet. And that was actually a really nice segue, Ellie, into what I'm about to talk about, so thanks for that. Before I start, I'd just like to acknowledge the traditional owners of the land on which I live and work and am presenting from today, the Ngunnawal people, and pay my respects to elders past and present. So, you may have heard in the media that in fields like social psychology or medical sciences, or more recently, behavioral economics, they're going through a replication crisis. And what that's referring to is the fact that when researchers were trying to repeat the same methods of a previous study, again in a new study, they were unable to get the same results, or maybe those results weren't significant. And these were papers that were coming from top journals in the field. And this is scary because it means that things that maybe we thought we knew, we might actually not know. And it's unclear whether we should continue to do research using these ideas that might end up not being true. Now, in ecology and biology, there's no equivalent investigation that exists, but it might also be happening in these fields as well. And that's because evolution and ecology research isn't immune to the wider issues of scientific research that might cause these problems. So in one of my favorite papers on this topic from Hannah Fraser, she asked, I think it was more than 800 ecology and evolution researchers whether or not they'd committed questionable research practices just through the process of trying to get through a review process and get something through publication. So here this plot just shows proportion. So some amount of people had said that they had not reported variables or covariates. They changed a hypothesis after knowing what the results were to be better suited. They'd rounded their p-values, maybe they excluded some data, they've added some data. And although they're unintentional, it appears that this is prevalent. And when combined together, this can make it really difficult to replicate the same results when you try to do it again with new data in a new experiment. But what about trying to reproduce the same results given the same data and the same analysis tools, which tend to be code? This paper actually came out like 2 weeks ago, and what the authors did, this is the top line here on the bottom, was they looked at about 370 papers in the top journals in ecology, like Ecology, Letters, Nature, and Science, just to check whether they made their data available. And what they found here is that this right bar shows that 80% of them did make their data available, which is actually really good. That's good news. It's a lot better than about 10 years ago. The problem is that this bottom paper in that list below did some research into whether you could use that data again to get the same results in some of these public data archiving or these top journals with these really good public data archiving policies. And by that, I mean, they really need you to make your data available, and it needs to be accessible in some way. They have strong policies on that. And what they found was that less than 50% of them were able to get the entire analysis results using that data that was shared. And the reasons for that were that maybe there was missing data, the metadata was incomplete, maybe it was aggregated in some sort of way, so you didn't have the raw data, or maybe it was shared in some sort of file format that wasn't very usable. And so even though the data is being shared, it isn't always reusable. And I'm sure maybe you've experienced that yourself when someone shared data with you. Now, this was just one half of the plot. The other half was investigating whether those same 370 studies or so shared their code. And this bar on the right shows that only about 20% shared their code. And the thing is, without knowing how someone cleaned and edited their data, even with the data, reproducing a result is very difficult. And I'm sure, again, that in many of our experiences, if someone shares the code with you, you might not get the same result. It might not even run. And so what we're finding out is that reproducibility is very hard. And I think we probably all have this sad, skeptical face that the snake seems to be making, feeling the same emotions as we hear that news, thinking about how hard it is to get someone to reproduce the result on their machine because the data needs to work with the code or analysis tools to work on someone else's machine. And when data and analyses can't reproduce a result, it creates something that's been referred to as research waste, or at least it was by the authors of this paper below. And what that was referring to in an academic context was that the process of making a whole, getting the results for data involves getting materials to do the research, make some methods, go and get data, get the results. But the problem is that a lot of the time, the incentives when doing these projects aren't on longevity. You know, a lot of projects are funded by limited project-specific funding, or maybe a grant is a short-term grant, and the incentive is on getting results, but not necessarily on longevity. And when that happens, it means that when we try to run it again, if we can't build on their findings. And often that means we have to start again from scratch to do the same things that were done previously. Now, it becomes an important question in that case to ask, how can we improve research reuse in ecology and biology? Now at the ALA, as you probably have heard a few times from the speakers before me, we acknowledge that this is an issue. And so I'm going to go through some of the things that we at the ALA work on to support research reuse. Now, actually, Ellie used a form of this before, so you might recognize it. I guess this one's slightly more exciting because it's beige. But I think it points out some of the main points that we've been talking about before, Peggy and Ellie before me, that there's a whole lot of data, especially in the last 20 years, and even more so in the last 10 years, coming from a number of sources. And this is mainly driven by human observations. The crux, though, isn't that this data, it's a competition between these sorts of data. The idea is that these are all now coming from more and more data providers. And I think the ALA standardizes and merges something like 3,000 different data projects coming from all these different sources. And so it's very important for data to be merged together in some usable way and be made open so that way it can be used for monitoring into the future, because a lot of data is coming in at the moment. Something else the ALA works on is a project called EcoAssets, and EcoAssets aims to make aggregated version data sets to support environmental reporting, like the State of Environment Report. And by doing that, the way that it does that is by merging data sets like from the Atlas of Living Australia, the Terrestrial Ecosystem Research Network, or TERN, and the Integrated Marine Observing System, or IMOS. So a lot of different types of data. And what the version part means is that you might have data set from April 2021, and then you might get new data and have a new data set from August 2021. Maybe you'll have another one in 2022. But if you want to go back and run the analyses that were done using the data from 2021, you can do that, and you can get exactly the same results. So this is increasing the reproducibility of these sorts of reports. Now, the extra cool thing is that if you look on the bottom right, there is this two different data sets from eco-assets. There's one that's used for monitoring, and it's more grouped by surveys. So you can kind of think about survey effort as something you might be able to see, which I'll show you a bit later. And then there's another one that has observations. So you might get things like species distribution information from that. And the really cool thing about that is that it's been linked to a threatened species list and endangered species list. So it's very useful for monitoring. Now, one of the final things that I'll talk about is these projects that I personally work on and a lot of our team work on to support good coding practices, namely the Gala package, which is now in R and Python, and also Ely Labs, which is a website that has how-to articles to show people how to make things themselves, like analyses or visualizations. And the goal for things like Gala is to help people who are coding write queries to download data from the ALA in ways that are easier to write, easier to read, and easier to repeat. And for ALA Labs, the goal is to show people what to do with the data once they have it. And here you can see a few examples of some articles we have. And the idea is that it shows you that you can do it on your own computer as well. Now, to just sum up, I wanted to go through some of the things that you might be able to do with the tools that I've talked about just before. And the idea is that I'm going to share some of the code with you so that you can hopefully do it yourself. So before I get to this bit, I want you to notice that on the bottom right, there's a QR code that shows it links to the code that it was used to make this plot. And the next few examples are going to be able to, you're going to use the QR code to be able to get them as well. So if you're interested, please scan that and you can open it yourselves. This beautiful data viz was made by our colleague, Dr. Sandhya Balasubramaniam, and it shows the number of survey events from 2010 to 22 using that eco-assets monitoring dataset that I talked about before. Now, what you'll notice is that as each year ticks over, there are some regions that are very dark green, showing that there's a lot of surveys there, and some that don't get green at all. And the value of this is being able to see whether there are certain areas that need to be surveyed more often, but also being able to see where there are certain areas that there might be more data, so you can ask more specific questions about a species of interest. Now, this example was made by Amos Smith, who was an intern recently earlier this year. He wrote an ALA Labs article about how to make this exact plot. So if you're interested, you can scan the QR code. But Amos was very interested in understanding and exploring where dingle observations were in the Atlas of Living Australia. And what he found was that there are quite a few in the Northern Territory in South Australia, which is what we can see here. And he was interested to know whether there was more observations in protected areas versus non-protected areas, and what he found was actually it was about 50-50. But this was made using Gala and with ALA data, and you can make this yourself by looking at that article. Now, finally, this is from a project I was lucky enough to be a part of recently. This was from a paper that's currently under review, led by Dr. Aaron Roger. And I think this kind of sums up a lot of the ideas that we've been talking about in this entire webinar quite nicely. So here we have 3 species and their species distributions with these hexagons. And in the green, that indicates that in that area, the majority of records were coming from citizen science. And in the purple, that indicates that a majority were coming from non-citizen science, so probably more like monitoring or standardized research projects. So here on the left, we have the shingleback skink, and you can see that a lot of them are purple, whereas in the middle here, we have Perrin's tree frog, and thanks to initiatives like Frog ID, about 40% of them are now coming from citizen science, or majority citizen science records. And now in the far right one, we have the red-browed finch, and thanks to eBird, pretty much all of them are green, and that's because it's really changed the game for observation. Now, the point here isn't to pit these two against each other, but to say that together, these are more useful than they are apart. And by standardizing them into one place where you can share them openly, they become very valuable, because these are just 3 examples. I'm sure you can imagine there's a lot of variation for many of the species that people are interested in monitoring. And by putting them there openly so people can use them and updating them regularly, this makes them more valuable. Now, in summary, reproducibility is very important because it allows people to reuse data in code to build knowledge. And by not allowing to, or having problems reproducing things, that can create research waste, which isn't very helpful for us. The ALA supports reusable research in ecology by aggregating and standardizing records from nearly 3,000 data resources. It also works on projects like EcoAssets that offer version data sets for environmental reporting. And it supports good coding practices like Gala, through things like Gala and Ely Labs, and I encourage you to check those out if you haven't already. Thank you.
Well, thanks, Dax. I think everyone's really enjoying this. There's good comments we're getting. And we've got a few questions to go through. So to start off with, we've got someone submitted 2 questions in one. The first part of it is, how often is ALA's database updated? That might be better for you, Peggy, I'm thinking.
Yeah, that's me. It's a complex question, but we run the index every day. It depends. There are probably around about 100 data sets that we check on every week and run an update if there's data, and then there's the rest that kind of just sits there and stays the same. But then we've got the difference between incoming data, and then we might have changes to, say, sensitive species or conservation species, and then we'll update those as required as well. So we'll reprocess the data as needed.
Excellent. And then the second part of that question is, in terms of plant pests, how do you coordinate with the Australian Plant Pest Database hosted by Plant Health Australia? Is that something that we do at the moment?
Let's see, we used to manage the APBD database. We do have a biosecurity project at the moment where we've got quite a lot of engagement with them, but I notice, so I can't speak to that exactly, but we are working with some data providers who are contributors to the APPD, like the New South Wales DPI, for example. So I think, they're working on how to open up their data sets.
Excellent. Yeah, perfect. And then for you, Ellie, this might be Ellie or Peggy or a mixture of both of you. What are the differences in how data for a museum specimen versus a human observation would be processed?
I'll have a go.
Oh, either of us. Yep.
Well, I'll just give the data geek. The data geek approach is that we put them through the same pipeline. Probably the main difference is that we collections record is usually has a basis of record of preserved specimen, or genomics data is coming in as material samples. The only difference in processing that we do to that is twofold. One is that we map it to actual collections, so we keep a list of all the collections, and we will make sure that the right data ends up in the right collection. And I think the other thing is that we have an annotation service that so people can come and flag an issue on a particular record. That's a service usually used by collection managers. Will go and use that service to flag their source database, so that the record is corrected and loaded back into the ALA. And
I think my contribution would be just to say that oftentimes with human observation data, particularly coming from something like iNaturalist, where it's just sort of, oh yes, I saw a galah. So you're not out on an organised survey necessarily, but from eBird you might be actually out on an organised survey. So the data from something like iNaturalist, what we get coming into the atlas is all the data that's known. So the species name and the locality data and the time data might be, and the photo that you take might be all that's known about that observation. Whereas with collections data, so museum or herbarium data, there's going to be a lot more data that's actually collected but not shared through to the atlas. So the museum's collection database is going to be much richer because there will be information about, for example, whether the thing's ever been on display, whether it's where it's stored, how it's stored, whether it's been divided. Like for a specimen, you might have a skin, a skeleton, a skull, some tissue samples, some genetic material, and they might all be stored and managed differently. So there's a whole lot of different parts to a museum specimen that aren't necessarily reflected through to the ALA.
Perfect. Now let's see. For DAX, are there any risks that people should be aware of if they do choose to share the code they're using for tidying and analysing their data?
That's a good question. Well, I guess some of the main things to be worried about is accidentally sharing any API keys. So sometimes you get, little keys that let you access various data or passwords or something like that. It's pretty easy to accidentally do that. The nice thing, though, is there are tools these days that you can run before you even make that code. If you're planning to share that in some place where people can access it, I know that R actually has a function that deliberately will find that data and remove it. So yes, there is a risk to accidentally share passwords, things like that. I think there's also a risk to sharing locations of species. So I think you need to be aware of that before you choose to share. But in general, if you are sharing data, there are tools out there that make that very easy these days that I guess maybe it's worth being aware of.
Cool. We've got one here. I have a question about copyright. When a new species is published, who holds the copyright of the species name and the description that is part of the publication?
So that's an interesting thing. So for something to be copyrightable, it has to be a creative act. Now, naming a new species, you could definitely argue would be a creative act, and the publication will definitely be in copyright. Usually, the copyright holder would be is usually the authors of the paper, but may be the publisher of the paper. So when you go to publish your paper, you may be signing over your copyright of that paper to the publisher. A species name itself is generally not considered to be a copyrightable thing. It's a piece of data, and you wouldn't normally be able to claim copyright over the species name. Species names are also things that change very frequently as new science is done and updates and more understanding is gathered. So it's not, we generally wouldn't think about copyright specifically in terms of species names, but we would in terms of the publication that the name appears in, if that helps.
Perfect. Yep. And we've got sorry, my screen logged me out, so I'm looking at the questions just in a bit of a different way than normally. So someone here has written they're a PhD student and code reproducibility is very interesting to them. So where could they learn more about that?
That's a great question. There's a lot out there. I think, honestly, there are lots of interesting things. If they're a PhD student, I guess they probably would have access to look at some journals. There's been some really interesting things coming out in some of the top journals about reproducibility, because as I mentioned, it's not just ecology and evolution. It's most fields are starting to take this really seriously. That's a nice place to get some more interesting things. I think when it comes to learning how to do it yourself, I think that's kind of the intention of things like ALA Labs, is to show that it's easy to make something reproducible. So, it's worth checking things like that out. But yeah, luckily there's a lot of things to learn from these days. It's not hard to find. Yeah.
We're just running out of time, so I'll end with this one for Peggy. So you mentioned the different search tools that we offer at the ALA, and there's a lot of them. If someone's new to using our website, what might be a good place for them to start if they wanted to have a go at searching and accessing data?
Probably our knowledge base, to go to some of our just our help articles and have a read through those, or look at our very excellent videos that you've produced, Juliet, for for how to do some basic searching if you're just starting out.
Perfect. Thank you so much. That's been really, really interesting, and we've had good feedback from everyone. A recording of this session will be made available over the next couple of days. So if anyone watching has a friend who might be interested or just really wants to deep dive and listen to it again, keep an eye out on our YouTube channel. We will be sending out a feedback survey to everyone who's registered for these events. So if you could please fill it out. It doesn't take too long, and it just helps us to understand what types of topics people are interested in hearing about so we can do more. And also don't forget to check out our last event we're running tomorrow for National Science Week. You'll find information on that on our socials or the ALA blog. And otherwise, yeah, thanks everyone. Have a good day. See ya.